Hello everyone, I'm Sanu Arora from the John Innes Center. The focus of my research is to understand the genetic basis of disease resistance in plants. In this presentation, I will talk about rapid cloning of resistance genes from wheat landraces and wild relatives. Today, wheat provides about 20% of the calories and protein consumed by humankind. Especially in the developing countries, wheat is a major component of people's diet. The pie chart here shows the wheat production in different continents. The sudden disruption of this supply can lead to famine and civil unrest. Diseases and pests constitutes a major limitation of wheat production, diminishing the world's projected harvest by an average of 21% every year. Their sudden and unexpected appearance can rapidly cause an epidemic, leading to large-scale regional crop failure. There are different control measures that one can take to protect the losses to diseases, or at least minimize them to have stable and predicted production. Control measures include use of pesticides, but frequent and excessive use of these chemical compounds develop resistance in pathogens against them. An economical and environment-friendly way to control disease is the use of genetic resistance, and it is especially important for those farmers who lack access to pesticides. To find new sources of resistance, it is very important to understand the genetic architecture of disease resistance. The genetic components involved in defense mechanism can be broadly characterized into qualitative and quantitative resistance based on its inheritance pattern and the effect size. The qualitative disease resistance is generally conditioned by a single resistance gene, recognizing a virulence factor in a classic gene-for-gene mechanism, conferring near immune response to pathogen. The large phenotypic effect makes these genes more amenable to detection and further molecular analysis. The picture here shows the number of years it used to take for cloning a single resistance gene, followed by several years for integration into a cultivar of choice. This was the timeline about 5 years ago for cloning qualitative genes. And this is the current timeline for cloning these genes. Here you see that there is not only a reduction in the amount of time spent in cloning a single gene, but you can clone several genes in a very short span of time. This can be achieved by the method that I developed in my postdoc research. This method is called AgRenSeq that combines association genetics with enrichment gene sequencing. To implement AgRenSeq, we phenotype a crop wild diversity panel with pathogen of choice. At the same time, we enrich the genomic fragments of each accession using an NLR gene-specific bait library, followed by sequencing of those fragments. Once the sequence and the phenotype data are both available, we are ready to associate them with each other. I implemented this method in Agelops torchii, the D-genome progenitor of bread wheat. Agelops torchii is divided genetically into two lineages, indicated by red and blue color here. We phenotyped the lineage 2 with 3 stem rust races in collaboration with Brian Stephenson's group at the University of Minnesota. The outcome was 4 clear candidate genes associated with these races of stem rust. 2 of these genes were positive controlled, SR33 in the first plot and SR45 in the second plot. The other 2 were uncloned genes, SR46 and SRTA1662. The SRTA1662 gene is a typical NLR with CC and PARC and LLR domains. To functionally validate this gene, it was transformed into wheat cultivar fielder, which was susceptible to this isolate. The transformation was done by Wendy Howard's group at the John Innes Center, and upon phenotyping the null and single copy transgenics, I confirmed that this gene was sufficient for providing resistance against stem rust. Another phenotype that we targeted in our group was stripe rust resistance in this panel. The panel was phenotyped with 7 stripe rust isolates from North America. The phenotyping was done in collaboration with Randy Kutcher and Guru Charan Barar from University of Saskatchewan and University of British Columbia, and Xianming Chen from Washington State University. Surprisingly, we got the same peak on chromosome 40 associated with all the 7 isolates. The peak underlies a cloned NLR gene. The association of this gene with all the stripe rust isolates indicated that this gene has not yet been deployed in agriculture. For all the 6 out of 7 isolates, the gene explained most of the resistance in the panel, except for one isolate where it explained 81% of the resistance in the panel. I reanalyzed the genotype-phenotype correlation for that isolate, this time with a subset of accessions which do not contain this gene. With this exercise, I found a second peak on chromosome 5D, which explained 17% additional resistance in the panel. In total, 98% of the resistance was explained. Since the development of this method, I have collaborated with multiple partners and helped clone about 12 high-confidence candidate genes in the last 20 months for different diseases. After completing the AgRenSeq on a wild progenitor species, we implemented this method on a wheat landrace collection available at GIC, the Watkins landrace collection. These Watkins landraces represent a snapshot of diversity from pre-industrial farming era from across the globe. We selected a subset of about 300 core lines and captured their NLR diversity using a bespoke bait library designed for this purpose. Because of the polyploid genome, it was more complex to set up this pipeline on hexaploid wheat panel. Very often, you would see false positive on homologous chromosomes. We explored genetically diverse Watkins panel for wheat blast resistance, a disease for which there is an urgent need to find genetic resistance. Wheat blast first emerged through host chump as a serious disease in Brazil and eventually led to an epidemic throughout South America. In 2016, wheat blast epidemics occurred in Bangladesh, posing a threat to the major wheat belts of India, Pakistan, and China. The wheat blast work was done in Paul Nicholson's group at the GIC. The panel was phenotyped with two different isolates of wheat blast. With the first isolate, we got a clear peak on chromosome 1D, which led to the discovery of an iconic wheat blast gene RWT3, which was involved in the host chump of this pathogen. The second candidate for this isolate was found on chromosome 7A. Both these candidates together explain 91% of the resistance in the panel. With the second isolate of wheat blast, the first peak led to the identification of RWT4 gene. This is another historic gene involved in the host chump. The B genome homolog of this gene was also involved in providing resistance for this isolate. A total of 83% resistance was explained by both these genes. You will hear the full story about wheat blast from Paul Nicholson in the next presentation. In this presentation, I have provided some examples to show how AgRenSeq approach can be used to understand the basis of qualitative resistance. The next big challenge is to develop methods for characterizing quantitative resistance loci. Because of the small effect of these QTLs, it is difficult to identify them with the existing methods. Taming this wild frontier of genetics will have profound implication for future breeding of disease resistance. Understanding both the qualitative and quantitative components of disease resistance will be useful to design gene-based markers to help breeders develop cultivars with durable resistance. In the end, a successful research program is not possible without funding and collaborators. Here I would like to acknowledge all the funding agencies who supported our work, people in Branderwolf's group at GIC, and our collaborators involved in stripe rust and wheat blast work. With this, I would like to thank you all for your attention.